Our team is doing final preparations to begin our main testing protocol on Friday. The testing being done this year is part of a multi-year study that Dr. Todgham has designed. The purpose of this experiment is to determine how juvenile Antarctic fish can handle the anticipated stress that they will be facing from changing ocean conditions. Dr. Todgham is interested in knowing how these small fish budget their energy demands. They can only eat a set amount, so how they spend that energy is important. Their body makes decisions. Do they grow and develop, or use energy to handle stress? Do they accomplish both? The answer to this question will help scientists know how changing our climate, and thus the ocean, will affect the overall ecosystem. It is important that all conditions are stable in each tank before we begin. There is way too much at stake to have mistakes. Today, Mandy, Melissa, and Andrew are doing water chemistries again for every separate tank in our system. They have been checking them regularly since they set up the tank weeks ago. The carbon dioxide system is very complicated. First, all carbon dioxide and water vapors are removed from the air. Then, specified volumes of cleaned air and carbon dioxide are remixed in just the correct proportions. It's taken weeks to balance the system and get the tanks just right. Now the tanks are stable and we have the fish. Time to start testing. We have two variables, water temperature and carbon dioxide levels. The criteria to establish these levels comes from predicted ocean conditions. The water temperature we are using are minus 1.3 degrees Celsius and positive 2 degrees Celsius. Melissa is taking the water temperatures to make sure each tank is accurate. They have been set up for more than 2 months, so we should be good. Andrew and Mandy are collecting water samples to be used to determine the level of carbon dioxide. Calculating this is not nearly as easy as the temperature. Water samples are taken in the lab where they are run in 2 separate tests. First, they test the pH of each sample. The pH tells how acidic the sample is. Acidity is determined by the percentage of molecules that have their hydrogen ion separated from the ionic bond.
So the first thing we do is we take the samples that we have of our uncontaminated seawater from downstairs and we warm them up in a bath to the same temperature at 25 degrees Celsius. And then we take a pipette, which gives us an exact volume of this sample, and we put it into a cuvette, which is this fancy vial. And then we put it into this machine, which is called a spectrophotometer. It goes in the machine, and then light goes through the water sample and measures absorbance at different wavelengths of light, which shows up on that computer. So then once we have that baseline, we pull it out, we take this chemical called impresol, we take another pipette, which takes another very specific volume, a very exact volume, and put it into this water sample, put it back in the machine, run it again, and then using a bunch of equations later we can look at the difference between the no dye and dye sample and determine what the pH of each water sample is.
Next, alkalinity is determined. This test measures the buffering capability of the water. This is how much the water will resist a change in pH when acid is added. Specifically, it measures the carbonate and bicarbonate levels. When you combine the results of both tests, you can do some crazy complicated math to get the carbon dioxide concentrations. My team programmed the computer to do this calculation for them, which makes the whole process much faster. That's why I have all my students learning to code. Scientists, as well as lots of other jobs, benefit from being able to write code. Because this experiment involves 3 variables, temperature, carbon dioxide, and time, we need to set up a complicated matrix of tanks. First, there are 2 big tanks that are each set at a different temperature. Then, within those temperatures, there are 6 sub-tanks with 2 different carbon dioxide concentrations, 3 of each. It is important to have 3 buckets of each condition to rule out any effects caused by the bucket itself. Each bucket holds 37 fish. We need to have that many fish for the data to be significant. Dr. Todgham adds a few extra fish to each tank just as a precaution. The testing protocol is very complicated. We are running all types of tests on day 1. Then in 2 weeks, then in 5 weeks. The first set of tests provide a baseline. We need to establish their activity and stress levels before they have a chance to show any effects of their environmental change. It is important in any test protocol to establish what normal is before you make any changes. This is called your control. You need to have something established to compare results to in order to see if there is a change. The last set of tests will be run 5 weeks from the initial setup. We are running a large variety of tests that will help determine if the fish are handling multiple stressors at the cellular, behavioral, physiological, and metabolic level. We conduct these tests over time because we think it will take time for them to adjust, if they are able to at all. The data will be taken back to UC Davis to be analyzed. It is important to note that in all of these tests, Dr. Tajim has taken care to ensure that the fish are not excessively stressed. We need them to remain healthy. All of our protocols are overseen by multiple agencies at the university level and above that regulate humane treatment of animals. She has special permission to do her research. After the main test, Dr. Tajim and her students will run extensive tests with the fish tissue back home at UC Davis. They will use these tissue samples to get a full picture of the physiology and the physiological changes that these fish undergo. They are careful to take only the amount of fish that they need for their tests.